Hello, my name is Alexandra, and I'm going to present to you my project called Comparative transcriptomics for identification of key genes among xylose fermenting yeasts through interaction networks and co-expression analysis. The use of fossil fuels to obtain energy is responsible for a large part of the greenhouse gas emissions in the atmosphere. Second generation bioethanol appears as a great option because it is a cleaner and renewable source and does not compromise food security. The main organism used in the fermenting process is Saccharomyces cerevisiae. However, wild strains do not have a metabolism for xylose consumption, which can be viewed as a productivity problem because xylose makes up 30 to 40% of the post-treatment material used. So, strains were genetically modified to allow this fermentation. So, the goal of this project is to compare the metabolism of xylose fermenting yeast with the insertion of the XRXTH and XY pathway through differential expression analysis, gene interaction networks, and co-expression analysis. The data for the XRXTH pathway was taken from a microarray analysis performed using three fermenting conditions. 1. Only glucose medium. 2. Mixed sugar medium. And 3. Only xylose medium. The data for the XY pathway was obtained from an RNA-seq with four collection points structured throughout the fermentation in order to encompass all phases. 1. Consumption of glucose. 2. Transition between consumption of glucose and xylose. And 3. Consumption of xylose. And 4. End of fermentation. So here's the project pipeline. For the network analysis, we took the differential genes at 2 points in order to contrast main consumption of glucose and main consumption of xylose. We used the F-string in Cytoscape to generate the networks and Encode to create submodules. And then we used the network analyzer metrics to identify hubs from both networks. We used double GCNA in R to make the co-expression analysis and define key modules. Then we used the gene significance and module membership metrics to verify if the hubs found in the network analysis were in fact significant in the co-expression analysis. If the genes passed our threshold, they were considered key genes. So here are our results for the network analysis. We obtained an XRXTH interactome with 485 nodes and an XI interactome with 571 nodes. In this table, we can compare the network metrics for both interactomes. For the XRXTH results, we found 8 submodules with Encode, and here I chose the main 4. And we use the data from the differential expressed genes to check which module is more important during the consumption of glucose or xylose. Here in red, we see genes with negative log FC, which means mainly expressed during the consumption of xylose. And in blue, genes that have positive log FC, which means mainly expressed during the consumption of glucose. In this side, we see the results of the co-expression analysis. We generated this heat map of the module sample relationship and we used the star to represent the key modules. Here are the key genes we found in our pipeline. We found a total of 8 key genes for this pathway, and I selected the main ones important during the consumption of xylose. So we have the FBP1 involved in glucogenesis and the metabolism of reactive oxygen species. We have the UBC5 involved in protein polyubiquination, and the ALD3, whose expression is induced by stress and repressed by glucose. For the XY results, we found 9 submodules using ENCODE, and here we have the main 4. Here again, we have the heat map with the module sample relationship and the key modules marked with the star. And we have the key genes. We found a total of 10 key genes, and I chose the main 3, which are GAD1, involved in cellular response to oxidative stress and glutamate catabolism, CYB2, involved in lactate metabolism, and CDC19, which is a kinase that catalyzes the final step in glycolysis. So thank you so much.